Good afternoon, everyone. I hope you have all found your seats. My name is Andrea Loy Mendoza. I am Program Manager for Roatan Marine Park, in charge of overseeing our research operations. On behalf of my team, the whole RMP team, I would like to thank you for being here. I would also like to thank all the people that we have online joining us via Zoom as well. Thank you so much for being here. Thank you so much for joining on this opportunity to share a little bit of our research endeavors, and we're also going to be introducing our partners in this very important project. Before we actually start, I would like to introduce my boss, Executive Director Bessie Aspra. Come along. She's going to be giving an introduction and some welcoming words. Thank you, Bessie.
Thank you, Andrea, and thank you all for being here and joining us here at RIMS. We want to thank AKR and RIMS, and also for all of you that join us through Zoom. I bet we all wonder if our coral reefs stand a chance. I think it's a question that we all have been thinking for the past, 10 years. And let me tell you that considering the threats that our reef ecosystems face every day, globally, regionally, and locally, we are here to give you some good news. There is a committed group of people working together, joining forces to keep our reefs in a healthy state. And I know that some of you and most of you have a part to share and have also the conscious that we need to protect these ecosystems. RMP has been working for over 19 years with other partners and also within the co-managers of the Bay Island National Marine Park. We're committed with a group of people committed to preserve these ecosystems, not just coral reefs, but mangroves, seagrass, and all our biodiversity here in the Bay Islands, of course for Honduras and the Mesoamerican Reef. We are a committed group of scientists, people, common people also that come and work with us as volunteers. And today we have the opportunity to meet one of our greatest partners. We've been working together for over a year. And I want to introduce our CAS team, the California Academy of Science. They will introduce themselves in a minute, but let me tell you that we are part of a big, huge global effort under a Cordap initiative. So we would like to share with you what we've been doing during this year. And of course, you know what we've been doing for the past 19 years. And this, what we have today, is the effort, not just as my team day, but RMP, different teams that have been working during these 2 decades. So thank you for joining us, and we will be hearing the details that we have under our research program.
Thank you so much for that, Bessie. And once again, thank you, everyone. We're very excited to have you here. And I'm even more excited to introduce the people who are going to be covering a presentation. First, we have Dr. Rebecca Albright. We also have Dr. Elora Lopez-Nandam. And we also have Lisa Larkin. All 3 of them are members of the California Academy of Sciences. We also have Steve Young over there. So they have been with us for close to a week at this point. We have been doing some exciting activities, achieving some important milestones, continuing to gather information for our project. So I'm very happy to formally introduce them. And now a proper round of applause for our presenters, please.
Hello, everyone. My name is Lisa Larkin. I'm an animal care manager within our Steinhardt Aquarium, which is actually something we'll talk about in just a second. Thank you very much to the Roatan Marine Park for hosting us. It's been a wonderful week. And of course, thank you to RIMS and AKR for hosting us today. just to quick overview, title slide. We're uniting research here for coral reef restoration. Thankfully, we were granted, thank you, the Cordap grant, and we'll be talking about that throughout this presentation. So just to give you a rundown of what the California Academy of Sciences is, this organization actually started in 1853, and it is a natural history museum, which you can see is this a pointer? Our dinosaur over here, as well as a planetarium here. A Steinhardt Aquarium is where both Steve and I work, as well as we have a research division called the Institute of Biodiversity, Science, and Sustainability, and we'll be calling that IBSS from now on. It's a mouthful. And this encompasses as well our collection of specimens, which is about 46 million specimens total. There we go. So this brings us to the collaboration within the umbrella of the California Academy of Sciences, which includes IBSS and Steinhardt Aquarium. I'm lucky enough to work with these incredibly intelligent women that you'll meet shortly. This is Dr. Rebecca Albright here and Dr. Elora Lopez-Nanda. This project started in 2018 and really started in a small room with very few corals, but we were able to spawn, and we actually have had successful spawn every year since the lab started. So our goals have actually changed over time. Initially, it was just looking at coral genetics in aquarium spawned corals, but Elora will tell the story about how this has quickly changed to encompass the Roatan Marine Park. So now our goals are as well as to advance conservation breeding and coral restoration science. I'd like to now introduce Dr. Rebecca Albright, who will be continuing our presentation.
Hi, thank you all for coming today. We're so happy to be here. My name is Rebecca. I'm a curator in the IBSS division of the California Academy of Sciences. Like Lisa mentioned, that's our Institute for Biodiversity, Science, and Sustainability. I'm a curator and I've been there for about 8 years, and my background is in coral biology and reproductive biology. I've been doing coral spawning work for about 20 years in Florida and Australia and Panama and kind of all over the world. And when I joined the Academy in 2016, it was the first time in my life that I had, or my career, I guess I should say, that I wasn't living proximate to coral reefs. And so as a coral biologist, I was a little challenged with how to approach that. But lucky for me, I was working in the same building, California Academy of Sciences, as the Steinhardt Aquarium, which is a professional aquarium, an AZA accredited institution that is home to one of the largest and deepest coral reef exhibits in the world. And so I approached the director and I said, I'd really like to establish a lab to study corals in human care and try to spawn them in human care here in San Francisco. And he was very supportive, and we tried it as a pilot project, which I'm going to show you a little bit about how that lab came to be. And now that we have an established lab, since we're not proximate to coral reefs, we want to utilize the science that we're doing there to make a meaningful difference in places that are proximate to coral reefs, which is how this project came to be. So the Coral Regeneration Lab, or CORAL, which we thought was a fabulous acronym when we made it, and then it actually gets incredibly confusing whether you're talking about the lab or the animal. So sorry about that. So for those of you who are not familiar with coral biology, corals can reproduce asexually and sexually. Most coral species, about 75% of coral species worldwide reproduce sexually once a year. through synchronous spawning activities, where they release gamete bundles, so these little bundles that are egg and sperm, into the water column. There's a sperm packet at the middle, and it's surrounded by eggs that have a lot of fat in the eggs. So when they're released from the coral animal, the fat makes them float all the way to the surface of the ocean. And if enough corals spawn, when those break apart, you have a flurry or a slurry of eggs and sperm in the water column. They fertilize, they create little coral larvae that can then go and swim and metamorphose and start calcifying on the reef. So this is the main way that we get genetic diversity on the reef. And in an era of rapidly changing environmental conditions, we want as much diversity as possible. And so we care very deeply about the genetic diversity of these systems on the reef, and we want to do everything we can in order to support it. So there's a variety of different cues, a hierarchy of environmental cues that go into triggering or eliciting this once-a-year coral spawning event. So all of these things in the literature have been suggested as being meaningful in terms of supporting coral spawning. So corals have to experience changes in seasonal temperature. They have to experience summers, they have to experience winters, and we think that it is the warming, the oogenic cycle, or like the production of eggs in many species takes 9 months. And we think it's the warming of the waters towards the end of summer that triggers the maturation of those eggs so that they're ready to be fertilized. So if we're trying to simulate these conditions in the lab, we have to make sure that the animals are experiencing winters and summers. So these are the things we have to try to simulate in a laboratory environment. They also have to experience lunar cycles, so new moons and full moons. Typically, the spawning event happens in late summer, a few nights after a full moon, a few hours after a sunset. We always say corals are surprisingly romantic. So we have to try to mimic these conditions in the lab as well. And so I'll show you, we have a series of blue lunar LEDs that go from 0% intensity in new moon simulated new moon situations to 100% intensity when it's a full moon in our lab. And then they have to experience sunrises and sunsets, so we're changing the photo period. These are the primary ones that we change in our situation. You can see the new slide up here. There it is. And so this is what we tried to do. So we tried to build a lab where we could trick the corals into thinking that they were experiencing seasons and lunar cycles and sunrises and sunsets in order to try to get them to be reproductively viable in the lab. And the way we do that is a series of controls here that basically I'm not sure if you can read this, but basically every month of the year, we program in the sunrise time, the sunset time, the moonrise, the moonset, the temperature. We have historical data from where the corals were collected. And every single day of the year, those conditions in the lab are changing iteratively to try to get these animals to think that they're experiencing these things in the wild. And I should say that we didn't originally develop all of these techniques. Most of this was developed by a researcher in London who did this at an aquarium, the Horniman Museum in London. His name is Dr. Jamie Craggs. And we were the second place in the world to do it. There's now quite a lot of places, aquariums, that are starting to integrate this type of technology. Okay, so this is a video showing you the construction of this lab. Thank you for your patience. This is just about a minute long. And there's some very terrible elevator music, so hopefully we can mute it. So we basically had to build a room within a room, like a photography, positive, a photography dark room, because we cared about light pollution. And then we collected Acropora hyacinthus, an Indo-Pacific species of coral from Palau. And this is when it's incredible as a researcher to be partnered with a professional aquarium, because people like Lisa are very good at knowing how to keep these animals alive. So we shipped them across the ocean to San Francisco and then held them in this lab and hoped that they would spawn. And this is a video of one of the animals spawning. We did have, the first year we did it, we didn't know whether it was gonna work. This was back in 2018. Typically this species in the wild spawns about 5 to 7 minutes after the full moon. I thought that they might go early. So my team and I, and we didn't know if they would spawn the right number of hours after our simulated sunset. So my team and I were monitoring every 15 minutes, starting 10 days before the full moon, the simulated full moon. And we did that every day for 3 weeks, and got very tired, as you can imagine. And so I have little kids. I have a 2 and a 5-year-old, and I finally said, I have to take a day off. And so I stayed home with my kids. And that day, 15 days after the full moon, the animal care person who worked with me at the time called me and said, all 15 of them are spawning right now. And it was amazing. So it worked, and we have been successful in eliciting coral spawning every year since. Here's, again, this is a different species, Acropora mellipora. So we've now successfully facilitated 7 or 8 coral spawning events across 2 species, Acropora hyacinthus and Acropora mellipora. This is a 1-year-old individual of Acropora hyacinthus. These are some of our 2-year-old individuals of Acropora hyacinthus. And for the first time, when the RMP team was with us last December, these F1, the first generation hyacinthus individuals that we had spawned and reared in our facility, spawned as well with their parents and with their wild counterparts. So we were able to fully complete the life cycle for that species, which was really exciting. I guess that's it. So that was really exciting. And so we're continuing to build on this. Like Lisa said, the main goals of this program, the Coral Regeneration Lab at the California Academy of Sciences, have changed a little bit over the last 5 years. As some of you are very well familiar, a lot of reef restoration work that happens in the wild is done through coral nurseries that are ocean-based nurseries. And in Florida last spring, unfortunately, the heat waves that happened in Florida, at many of those nurseries, there was 100% mortality of those ocean-based animals. And so the goals of this facility have changed a little bit in an unfortunate way of really trying to move the needle in terms of land-based conservation breeding efforts in association with public zoos and aquariums in order to have a land-based genetic rescue and refugia for animals that and establish best practices for those types of programs. And then we are also, as we'll tell you about, trialing a lot of techniques to hopefully enhance growth and survivorship of animals in the wild, do everything we can to make restoration more impactful, more successful, and then work with partners, these amazing people, in order to have meaningful on-the-ground impact. So with that, you'll hear from me again later, sorry about that, but I would like to pass it to Dr. Elora Lopez-Nandem, who is going to talk to you about the specific project we're working on right now with RMP and how it came into existence. Thank you.
Hi everyone, I'm Elora and I've been working with Rebecca Albright for over 5 years now, starting when I was a student, and officially joined Cal Academy about 3.5 years ago now. My background is in coral genetics, and so when I first learned that Rebecca had started a lab where you could reproduce corals, not in the ocean, but in an aquarium setting and keep them alive, I was so excited to be able to join and to be able to start asking these questions of, if you are going to start breeding animals in aquaria, what does that look like? Do you start creating animals that are healthy and able to go back into the ocean successfully? Or do you have to figure out how to change your protocols in order to produce animals that are going to be genetically diverse and that are going to be able to survive once you put them back out into the ocean? So about 1.5 years into my time at Cal Academy, I first heard about the Roatan Marine Park through a few of our colleagues at Cal Academy who are mesophotic divers. And so they had been coming to Roatan for a couple of years to do a bunch of training, and they were very excited about the reefs and about the people that they had met at Roatan Marine Park. And in the conversations that they had, they learned that Roatan Marine Park was starting to get interested in assisted sexual fertilization of corals on these reefs. And so they brought that information to Rebecca and Lisa and I and said, you know, you might really be interested in finding out what these folks are doing in Roatan. And so, of course, we said that we were. Most of you in here are probably quite familiar with Roatan Marine Park is and what they do, but they're a nonprofit organization established 19 years ago who works on all sorts of conservation activities as a co-manager in the Bay Islands. And so that ranges from patrols to coral restoration to all sorts of other types of monitoring activities. So when Lisa and I first came down here in September of 2022, it was in order to work on the first assisted fertilization of corals that was ever to occur in Honduras. So this was a really incredible workshop that was organized by Roatan Marine Park using information that they had learned from Sea Corps International about how to collect gametes from corals as they spawn. So that means doing night dives, collecting those gametes, bringing them back onto land, fertilizing them, and then putting them out into what we call cribs, which are kind of these floating, kind of almost trampoline-like things, where you can get the little coral larvae to actually swim around in the water and then start to settle onto substrates. So as soon as the slides load, we'll look at what that looks like. So this is the team that we had in September of 2022. You'll recognize everyone in the middle standing in front of the CRCR at RMP. On the left-hand side, we have a team carrying the crib onto the dock where it was going to be brought over to West End to sit in the bay. And on the right side, you see everyone looking very proud of the coral spawning nets that we assembled together. And I just want to mention that this workshop that was put together by RMP featured a whole bunch of different collaborators across both governmental and non-governmental partners throughout Honduras. And so it was a really great opportunity for people to learn from each other and to really start to understand why coral reproduction is really important for the health of reefs. So this is the magical moment. If you can imagine, this occurs at about 9 p.m. at night, about 25 feet under the surface of the water. For several days at this point, we had all been doing night dives and waiting and waiting and watching and wondering if the corals are going to do something. And then on this particular year, they in fact started reproducing on Honduran Independence Day. So that was very exciting. So they had their own little fireworks under the water here. So once that happened, not for this coral, but for many of the others, we would put the nets on top of the coral. Those gametes would then start to float up through the net and into a little tube at the top. And then it's pretty easy to just take the tube off and cap it underwater and then bring that back up to the surface and have all of your coral gametes ready to go and get fertilized. So while this was really incredible, the other really important thing that came out of this first collaborative project between Cal Academy and RMP in 2022 is that Lisa and I were able to sit down with the executive director at the time, Francis Lambu, as well as the program manager at the time, Gabi Ochoa, and start asking them and talking to them about what their priorities were for the Roatan Marine Park and whether there were places in which Cal Academy and RMP could work together to facilitate those different priorities and goals. So some of the things that were discussed and prioritized were designing a coral rearing facility at RMP. So a place where corals could be both reared on land and potentially experimented upon, and really enhancing the research capacity of Roatan Marine Park as well as their partners. Of course, to go along with that, we had to think about fundraising for said facility and how that would actually come about. As well as thinking about how build out of this facility would go and how training staff to use and maintain it would happen. And then starting to think about the future, planning for future spawns, including plans for monitoring, settlement, and really having a strategic quantitative way to measure how our efforts are actually contributing to coral cover on the reef. And then finally, there was discussions about a scientific diving course for RMP staff and potentially other interested partners in Honduras. And so I'm really pleased to be able to say that of these priorities that were identified in 2022, we have achieved the first two of them. We're in progress with numbers 3 and 4. Number 5, we haven't gotten quite to yet, but we're working on it. And so I say this all to say that we're really happy that this has been a really productive collaboration in the last 2 years, and we're really excited for what's to come. And so now we really want to get into what's called the Coral Accelerator Program Award, which was awarded by a G20 initiative called Cordap, which is the Coral Research and Development Accelerator Program. And so they first made an initial call for proposals in 2022, and the program manager at the time reached out to me. Truly, it was pretty serendipitous, probably about a week after Lisa and I got back to San Francisco from Roatan, and she said, I just learned about this call for proposals. We'd be very interested in submitting a proposal with you. And so Rebecca and I sat down with Gabby and we started talking about what could this proposal look like? What are the things that we would be really interested in working on together and what would be really meaningful for Roatan and for the Bay Islands? And so we're very happy to say that that proposal was funded last year in the summer of 2023. It's called Interventions to Improve the Health, Growth, and Survival of Larvae and Settlers for Sexual Restoration of the Mesoamerican Reef. And so we have 4 co-PIs, 2 from Cal Academy, Rebecca and myself, as well as Jennifer Keck, who is a board member at Roatan Marine Park, and Andrea Godoy Mendoza, who is the RMP program manager. This collaboration has also been incredibly supported by Carolina Rojas, who is a research coordinator at RMP, as well as Bessie Aspera, and so many other people at RMP who are really proud to work with. So now I'm going to bring Rebecca back up to tell us a little bit more about what the Cordap project actually is, what we've achieved so far, and what we're planning to do with the remaining time in our grant. Thank you all.
Hello again. So as Elora mentioned, we were privileged enough to be awarded this grant, which runs for the next 3 years. We started last October, and we will run through 2026. And when Cordap was trying to identify top priorities, really they were struggling with the concept that the pace of restoration and conservation obviously isn't keeping pace with the rate of degradation. And so there were a bunch of different priorities that were identified that they would fund or award these grants for. Some of them had to do with removing human bottlenecks for asexual fragmentation and planting corals back out onto the reef, like funding automation and technology in order to get robots out doing it, things like that. Some of them had to do with the one that we were targeting has to do with enhancing post-settlement growth and survivorship. So we already said that this spawning event, which is the main way you get genetic diversity on the reef, only happens once a year. And most of those individuals wind up dying. A lot of them are eaten. It's a huge predatory event when they're spawned in the wild. A lot of them, when the larvae actually go and settle and start growing, they're outcompeted, they're overgrown, they're predated upon. If the conditions aren't good for water quality or bleaching, they just die from external stressors. So the majority of those individuals wind up dying. So we can do the best possible, like we can get 99% fertilization in the lab, 70, 80, 90% settlement, and then we put them out on the reef and things are still very, very challenged. So one of their top priorities was how do we identify ways to enhance post-settlement growth and survivorship so that all of these efforts that we're doing yield more individuals on the reef. And so collectively, we thought of some ideas of how to do that. And so we identified 4 different culturing techniques that we wanted to try in order to enhance post-settlement growth and survivorship. And these are 3 of these are based off of the hypothesis that we know that so the eggs have a lot of fat and then they turn into larvae. And those larvae are typically non-feeding. They might feed a little bit in the water column, but for the most part, they are what's called lacithotrophic, which means they get a certain amount of energy when they're born and they just use it up. And so they don't get more energy by feeding in the environment until they settle, calcify, and then once they have these symbiotic algae inside of them, then after, this is usually around a month old, then they have another food pathway. And so we started to think, okay, well, if they are very stressed and they're using up this energy supply really quickly, then maybe they're not growing and surviving that well. But if we can alleviate some of those sources of stress and or offer them other pathways of food, maybe they can reserve some of that initial fat and food that they got when they were born and allocate it towards growth. Because we know that the faster they grow and the bigger they are, the better they survive. So we started thinking about ways that we can help them not use up their energy so quickly. So I have a background in ocean acidification, which is the changes in the chemistry of the ocean, the decrease in the pH of the oceans when the oceans absorb carbon dioxide from the atmosphere.
And we know at this point that those changes in the ocean chemistry make it really difficult for a lot of calcifying critters to calcify. So we have done some studies in the past that show that if you buffer the seawater, they actually calcify a lot more quickly. And so we started to wonder whether this was true for baby corals too. So if during that initial, when they settle and they lay down their initial calcification step, that is hugely energetically intensive. And we wondered, if you buffer the seawater, does it make it more favorable and easier for them to do that? And then they can put all that energy that they would have put into calcification towards growth and survivorship. So that's one of these interventions. Another one is giving them a different nutritional pathway. So we dose them with amino acids, which they can absorb, and see if that helps them grow better and survive better. We've tried inoculating the larvae with symbionts earlier in the life cycle than they might get them in the wild, to see if we can establish an alternate nutritional pathway earlier in their life cycle and help them survive and grow better that way. So those three are really looking at enhancing growth and survivorship through culturing techniques. And then, and these, I will say that these we specifically chose because we wanted to choose things that would be really easy and low cost, minimal resources to do anywhere in the world. We didn't want to do something that required a ton of money and a ton of resources in order to do it. So these are, we priced out what this would cost, and they're very low cost, minimal resources. And then Elora is really interested in selecting or speeding up adaptation to deal with bleaching. And so this final intervention looks at taking a culture of larvae, of coral larvae, and exposing them to very high heat temperatures, and like 36 degrees Celsius, and selecting, so some of those individuals wind up dying off. The ones that are a little more thermally resilient wind up living. And then we're rearing those out to see if we have effectively selected for individuals that are more thermally tolerant and are bleaching resistant, quote unquote. And so so we're testing these at the California Academy of Sciences with the, and then we are, one of the big deliverables during year one, which we're working on right now, is building a larval rearing facility at the Roatan Marine Park so that RMP is able to scale the sexual restoration work that they are doing. And the techniques that prove viable, that seem like they might be promising, we then want to trial down here at the facility that's being constructed at RMP, and hopefully utilize those techniques to increase growth and survivorship of coral recruits out on the Honduran reefs and outplant sites. So in December of last year, we hosted the RMP team, 3 members of the team. So we had Jennifer from Anthony's Key, Andrea and Carolina came up and spent about 3 weeks with us for our coral spawning event at the Steinhardt Aquarium. And we conducted the initial, set of experiments of these 4 interventions to see which of these might be viable. And so basically we spawned the corals in our systems like you saw a video of before, and then we created larvae. We all did this collectively as a team. And then we split the larvae. So we had some that were just control. We had some that we exposed to those different treatments. And we are currently rearing them all out. So they are 9 months old at this point to see whether it actually does enhance growth and survivorship of these baby corals. We use little settlement plugs, and then we image them every month in order to track them. And they're all, there's colored cable ties. This one's a blue cable tie, which is a control, right? A control. And so you can see this is when they were 5 days old. This substrate is 2 centimeters in diameter-ish. And on this substrate, you can see the little white dots are little larvae that have settled and metamorphosed. So this is 5 days old, and this is the same scale. So this is one of those settlement substrates at 9 months old. You can see that baby coral has grown and is now covering pretty much the entire tile. And then we have them cable tied, different colors, and we're tracking the growth and survivorship to see if these culturing interventions worked. And so far, there's 2 that seem promising, the buffering one and the heat selection one. And so these are preliminary results from the first 9 months. And what we have seen is that, oh, I don't know why this is over here. It should be right here. This should not say control stress. It should say heat stress. So that got a little lost in translation. Sorry about that. This is the controls. These are the amino acid treatments. These are the buffered treatments, and this is the heat stress treatments. So far at 9 months after, or at 9 months old, the recruits that we had exposed to buffered seawater during settlement and metamorphosis are showing about 356% higher survival than the controls. And the ones that were exposed to high temperature and selected for thermal tolerance are showing over 500% higher survival. And so we're very excited and surprised by this, and we want to repeat it obviously this year just to make sure that what we're seeing is real. And so I'm excited to say we have 2 members of RMP that are coming up this year for our spawn again in December, and we're going to repeat these and make sure what we're seeing is viable, and then the intention is to then do it down here in the facility that we're building. So we're entering year 2 of this grant, and the main priorities for year 2 are installing the facility that is already built. It's actually here on the island. And so construction and installation of that is slated for November. So hopefully if you swing by RMP after November, you could see that facility. And we should be actively working in it by June, hopefully, for the D-Lab spawn, the Diploria labyrinthiformis spawn. We're hosting the RMP staff in December to come up for trialing of that 2nd year's worth of experiments. And then we will be back next year for a variety of different coral spawning events, the Diploria labyrinthiformis, the brain coral spawning, the Acropora spawning in August, and then the Orbicella, the star coral spawning in September, to utilize the facility and try to enhance the outplanting of sexually derived recruits.
So that is all we have for you today. I think we're happy if there's time to answer any questions that you might have. Or Andrea, I don't know if there's we can mingle, or do you want us to sit up here and answer questions?
Thank you so much. Let's do a round of applause for our amazing presenters. Thank you everyone. Thank you so much, Rebecca, Lisa, Laura, for that presentation. We are extremely proud to be partnering up with such amazing individuals, such amazing scientists, professionals. As you saw from the presentation, we have already achieved some very significant milestones. We already have important data, and it's all very, very encouraging, especially because we're going to be able to replicate that in a country like ours. So we're extremely proud. This is something that we're very excited about, and this is an opportunity for everyone that is interested and cares about corals to get involved, ask us questions, and right now specifically, we will have space for a round of questions. One of the members of RMP staff is going to be going up to you with a microphone in case you have a question. Please just wait until we tell you go ahead, because we do have an interpreter present online. So if you do have any questions, please just raise your hand, and one of the RMP members is going to be going up to you. Before we start, I would just like to have the presenters come up here, and then I will be giving you the microphone to answer questions that may arise.
I was just curious if the simulation inside the lab with the lighting or, a new moon or full moon, you're simulating what is actually occurring in real life outside, is that correct? So when there is a full moon, you're simulating the lighting in the lab to be that as well.
Yeah, that's exactly right. And so we base it off of where the corals came from. So the corals that you saw came from Australia. So we plug in the sun. Those are austral, like southern hemisphere corals. So their summer is in December. And so we plug in their local temperatures, their local sunrise and sunset. So it's not necessarily that they're seeing exactly our conditions outside where we are, but they're seeing what they would see in the wild from where they're from. And we do know that in doing that, that they spawn at the same window as their wild counterparts. I will say the one exception is that we trick them a little bit because typically they spawn in the evening, as you know, and then if we're doing this work in the wild, we're up to like 2, 3, 4, 5 o'clock in the morning doing our work. And so we simulate sunset at 11 o'clock in the morning so that they spawn at 1 or 2, and then we don't have to be up so late. And that works. So it's nice for us.
All right, so, great.
Well, first of all, thank you so much for all your insight and everything that you shared with us today. I also wanted to ask, with the challenges that you were faced with the corals over there, with the spawning, what's your insight compared to the ones that we present here, with all the parameters and all that stuff? Or maybe we can leave that for later.
Yeah, no, I mean, I'll start and then I'll see if anybody wants to add anything. It is difficult to compare because these animals in the wild here are exposed to uncontrolled conditions. So we're able to control our temperature. We're able to feed them every single day. If they start to show signs of rapid tissue loss, Lisa helps them. She's an expert at watching each animal really closely and then knowing how to intervene. And so it is difficult to compare to the wild animals where it's obviously more difficult to control their local environments. And we have different challenges in our system. Keeping animals in human care presents a unique set of challenges compared to the wild. So I don't know. I don't think I answered your question. Does anybody have?
I think the question is an excellent one because I think it's exactly one of the things that we're trying to figure out over the course of this grant, which is how transferable and how applicable are these different therapies across different species. And so one of the reasons we thought that Roatan Marine Park was a great place for us to do this work is that in our lab, we're keeping Indo-Pacific species. And so to be able to test this on an Indo-Pacific species and then on different Caribbean species, so not just one species either, but acroporids and Diploria and Orbicella, we can start to see, does the buffering treatment help some species but not others? Or does the temperature experiment help some species but not others? To what extent are these types of therapies and methods applicable across a whole bunch of different types of corals, and to what extent are they not? So I would say that's something we really don't yet know.
And that's a really great point. And I'll also just say that it's a wonderful question that we don't know yet, but understanding when we keep these animals in human care, if they change in ways where they are really different than wild animals, is a really big question that we're asking right now, because obviously it doesn't do us the kind of good that we want if we keep them and we reproduce them generation after generation, but then you want to rewild them
and they can't withstand the conditions. And so I think understanding if we breed these animals on land and want to put them back out, are they fundamentally different than the animals that are out there, or are they able to acclimate? So those are all, it's a great question. We don't have the answers, but we're working on it.
Thank you so much.
All right, I have a question over there.
Yeah, if no one else is asking questions, I'll ask. There was some coral taken out of the Bahia de Tela that was taken to a research lab in Florida that apparently did well during the last few years in terms of ocean temperatures and so on. I was just wondering if there's any collaboration between your lab and their lab as to how or the research they're doing and if you guys sharing information.
Yeah, I think it's a great question. I was actually on a live-aboard trip last month in the Florida Keys with some of the researchers from University of Miami who are involved with that work. And so there is quite a community of coral researchers across the country and across the globe that we really are trying to share information with and tell people about, you know, these are the things that we're trying and that we're seeing. What are you trying? What are you seeing as well?
I don't know 100% what the goals of that project are. I'm not intimately involved with it, but my understanding, my limited understanding, I will say, is that they're trying to understand whether those animals are more stress resilient than some other animals, and then perhaps integrate that knowledge with some of these other practices. Like we do have areas where people are trialing, okay, can you take stress resilient corals and breed them with corals from other areas and produce stress resilient corals for these other areas. So I think that's kind of the logic and perhaps where that project is heading. I don't know, we are not directly integrating with them yet other than informal conversations. But I think the hope is that we'll all integrate and get there if it proves viable, yeah.
Are there any other questions in the room or in the Zoom room? No? One? Okay, we are always faced with questions when we are here on the island. And one of the most important questions that people ask us is how do we anticipate or how do we foresee that the achievements that we are able to achieve through this project will extend past the 3-year timeframe of the Cordap project?
We had a 2-hour meeting talking about this the other day.
So I think we collectively, not we, but the whole group is very actively thinking about early in year 1 about how we make this sustainable, because the last thing any of us want is to put this amount of energy and resources towards building a facility that is funded and viable for a couple of years and then isn't sustainable. So we brainstormed a lot of different ideas, I think, in that session that we're going to explore. But I think at this point, we're really just as a group, and we welcome, I should say, on behalf of me, I don't want to speak for anybody else. We welcome input and ideas from other people who want to be involved or have ideas about how to make it sustainable. But we really, I think the goal by the end of these 3 years is to have a model of what that looks like, of who has access to that facility, how the ongoing costs for that facility are managed and supported so that it's not just grant supported 2 years at a time. We want it to be capacity building and sustainable and serving the needs of the communities to rebuild the reef, genuinely. And so we're having those conversations early. If you have thoughts on how to make that successful, we welcome those. Yes, these are our, please email us. And we will be back, you'll see us a lot, I hope, over the next few years. And I didn't say this at the beginning, but I just want to, they've been here before, this is my first time here, and this is
everyone here has been so incredibly supportive and amazing and welcoming, and I think the best you can get is working on good, meaningful projects with an amazing group of partners and collaborators, and that's what we have with this project, and I'm incredibly grateful for that. So I just want to say thank you to RMP, to AKR, for everyone for hosting us this week, for making this a success, and we're really, really happy to be here and excited for the next few years.
All right. Thank you so much for your insights. We really appreciate it. You can take a seat again. Do we have another question? I'm going to ask you to please repeat that on the microphone so people on Zoom can hear your question.
It's on, okay. Just wondering how much it costs to do this on a yearly basis. How much is the grant per year? And you have a 3-year grant, so I'm just curious, because
So there's different parts to that question. So there's the initial, in year one, there's the initial cost of actually constructing the lab and installing the lab and all of the groundwork prep and permitting and everything that they had to do. So there were initial startup costs in year one. There's the ongoing staffing and personnel needs that we need both at the Academy of Sciences and at RMP. And then there's the portion that is ongoing support for the actual restoration, conservation, spawning work, which is the boat rentals and the restoration substrates and all of those types of things. So there's really 3 different pieces to that. I don't know how much if you want to speak to actual numbers or.
Does anyone else have any other questions? All right, we have a question from someone over Zoom. Thank you for that, Velvet. And they are directly asking how the local diving community can get involved with this project. I hope you don't mind, but I would like to answer that question. Awesome. So that is an excellent question, and we really appreciate the interest. As you can see, all of us in these colored shirts are part of the RMP, but that is not enough people to do the amount of work that is required to actually have successful restoration initiatives. So if you are a diver, a dive professional, someone who cares about the reef, someone who makes use of the reef, you are more than welcome to become involved. One of the most specific activities that I can think of now is getting involved with coral spawning monitoring activities. These are usually night dives. They take place after sunset. We have been in the water past 10 p.m. at times, depending on the species, but we need people to get in the water with us. We are willing, through this project, to teach volunteers how to identify corals, learn about coral biology, coral ecology, show you how to detect when spawning might happen, how to notice the changes not only on the corals themselves, but the environment, so that you are able to help us collect more and more information so that we can continue to validate our coral spawning prediction calendar for Honduras. Additionally, as Rebecca mentioned, we are really trying to brainstorm ways in which we can bring leaders in the community into this so that we can harness all of that potential that exists locally, so that they can be leaders of specific expeditions, be in charge of volunteers, know how to collect data, know how to schedule these types of activities. We really need those hands, we need those eyes on the reef. Also, for dive shop owners and operators and business managers, if there is any way in which you can support us logistically, we are always in need of boats, we need tanks, captains, fuel. We need all of these resources to continue to do the activities that will feed into this amazing initiative. Additionally, if I can't cover all of the potential for the local community to get involved, please feel free to reach out to us. We are on Facebook, YouTube, Twitter, Instagram. Feel free to reach out to any of us. We will surely find a way in which we can get you involved, but primarily being those eyes, our aid on the reef would be probably the key point. I don't know if anyone else wants to add anything to that. But yeah, we're open. All right, well, it has been an extremely productive session. I would like to once again thank Lisa Larkin, Dr. Rebecca Albright, and Dr. Laura Lopez Nandam for being here, for sharing their insights. So please, another round of applause for them. Thank you so much. Additionally, thank you very much to the Roatan Institute of Marine Sciences for allowing us to use this space as gracious as ever. We're really, really thankful. Thank you so much. Lovely space to be holding a presentation like this. We do apologize for the initial technical difficulties, but we really appreciate your patience and once again, your presence. On a longer term, we would greatly appreciate your support, your involvement, your engagement. Reach out to us. Remember these faces in these blue shirts. These are the people that you can reach out to. These are the people that you can ask questions. These are the people that can support you and that you can also support. So feel free to reach out. And without more words for you, we do have some snacks and beverages over at the back. So feel free to help yourselves. And once again, thank you very much for being here.